This is the 14th lecture of the semester, and we'll be covering signal transduction, the beginning of our segment on signal transduction in this lecture. The next three lectures will all be covering various aspects of signal transduction, and this is from chapter 12 in the text. And really, we try to give a good overview of some of the different major pathways in signal transduction, and we'll show some specific examples of uh, a couple important types of signaling molecules. Okay, and today what we're going to be covering is one, just kind of some general features of cellular signaling in general, and there are five really general features that are very important in all signaling pathways that we'll focus on. Okay, and that comes from the first section of the chapter. Okay, then we'll move on to section 2, and these are the G protein coupled receptors, or GPCRs. This is an important class of receptors and signaling pathways. And the specific example we'll see in this section is the beta 2 adrenergic receptor, or epinephrine receptor, or adrenaline receptor. Okay. And then we'll skip ahead to section 10 and talk about the signaling pathway involved in sight. And that also involves another G protein coupled receptor that we've mentioned before, and that is rhodopsin. Okay. And rhodopsin is the G protein coupled receptor, and then this is coupled to some ion channels, and we'll be covering that next time as a little bit more in detail, the ligand gated ion channels. Okay. So some biochemical roles of signal transduction are given here on this slide. Okay. And there's a number of different molecules that's in the body that can be used for signaling, antigens that lets your body know, your immune system know that there's something that needs to be attacked or removed that's foreign. Okay, you can have hormones that sort of act throughout the body, neurotransmitters that act specifically in nerve cells. There's also things like physical things like light that elicits a response in your body to actually view it. Touch is another signaling pathway. You physically touch something and a signal gets transduced from, let's say your hand all the way into your brain, that you can feel that. And also pheromones. Right. The signals cause changes in either the cell's composition or the cell's function. Okay. And some examples are shown here of that. There are thousands of signals, either outside in the environment or also within our body. Okay, but only approximately 10 different receptor types or patterns have been found. Okay, so you can see that there's kind of a limited number of receptor motifs, if you will, that are used to detect many signals. A list of some of the important receptor types are shown here, and this is given by the book. G protein coupled receptors. And first I should mention, receptors, when we're talking about receptors, we're going to be focusing on membrane-bound receptors. That's why this chapter is sort of right after our discussion of membranes. But there technically are some soluble proteins that are considered receptors, but we're going to be focusing on the membrane-bound ones. Okay, and when a receptor binds a molecule, what we refer to the molecule that it's binding is its ligand. Okay, when it binds, the receptor binds its ligand, it's going to exert some sort of a physiological effect. That's also known as an intrinsic effect. And the types that we're going to be focusing on in this class, one are the G protein coupled receptors, and we'll see a couple examples of those. Again, one is the epinephrine receptor or beta-2 adrenergic receptor, and the other one will be rhodopsin. Okay. We'll also look at receptors known as enzyme-linked receptors or tyrosine kinase receptors. Okay. Those we're going to be focusing on next lecture. Okay, some of the specific examples that we'll see, one is the insulin receptor and epidermal growth factor receptor, and then also something known as cyclin CDK. Cyclin CDK is a little bit different because it's not a membrane-bound receptor, but it's involved in cell cycle regulation. Right. Next lecture we'll also be talking a little bit about ligand-gated ion channels, and specifically the sodium channel. It says potassium here, but we'll be talking about the sodium channel specifically next time, and something known as the nicotinic acetylcholine receptor, and both of those are involved in nerve signaling. Okay, these are shown in gray because this slide is from a previous class that I did where we didn't cover these, but in this class there will also be the third lecture in this signaling series will be a very short lecture, and we'll be talking about a little bit about integrin receptors and some nuclear receptors as well. So here's a list of the 5 features of all signal transducing systems. This is figure 12.1 in the book, and we're going to be going through these sort of step by step and give some examples of each. Okay, so the first one would be specificity. And receptors are very specific for the signaling molecules that they bind. Okay, so there's really, primarily, if you're a good receptor, there's one signaling molecule that will bind, or one type of signaling molecule that will bind, and other molecules that don't have that correct shape can't bind in that receptor, and they can't elicit that response. Some typical ligands for receptors are things like small ions. The example, it would be in bacteria. There's an iron receptor. Organic molecules, these you see a lot in humans, and we'll be talking about the adrenaline or epinephrine in the beta-adrenergic receptor here shortly. It would be an example of an organic molecule as a signaling molecule or ligand for a receptor. Polysaccharides, so those are multiple sugar molecules attached in a chain. Peptides, small sequences of amino acids are called peptides. They can be signaling molecules, and an example there would be insulin. And then proteins themselves, and those are things like growth factors, are usually larger proteins, can be signaling molecules. Okay. The second feature of signaling transducing pathways would be amplification. And you can also think of this as being a cascade. Okay. So a signaling molecule will bind a single receptor. Okay, that receptor generally will activate multiple copies of a second enzyme or protein. Okay, so it will activate, in this case, and our receptor would be enzyme 1, getting that signaling molecule, a binding of that signaling molecule. Then it will activate a second enzyme, and in this case it activates 3 of them in this example. And then enzyme 2 then will activate 3 more of enzyme 3. So you can see that one signaling molecule can now have activating 9 enzyme 3s. Okay, so this is kind of a generic example, and we'll see a more specific example of this in a minute. Okay, the third feature, modularity. Okay, it turns out that proteins can form complexes with each other, these signaling proteins, and they can make really different complexes that can have different activities. So you'll see the same building block proteins in different pathways, and that's an example of sort of, you can think of it as trying to think of a good example, and nothing's coming to the top of my head right now, but if you have something that can be used in multiple ways, that, I guess that would be the closest example that I can come to right now. Okay, and phosphorylation is, these proteins being phosphorylated is a good point of interaction for these complexes. I don't know if we actually see very many specific examples of modularity in this class in the pathways we look at. I don't recall any off the top of my head. But if you think about it, if you have thousands of different signals and you only have 10 different types of receptor available, some of the secondary signaling from those receptors, it would help out immensely if you can use the same protein in different pathways and in different complexes. Okay, the fourth feature, it would be what's known as desensitization, or adaptation. Okay, and this is where you have a signal that activates a receptor. Then you have some sort of a response from that receptor, from that signal. That response at the end, once you have enough of that response, it feeds back and it shuts off that receptor. Okay. So you can think of this as feedback. And then finally, the fifth feature would be what's known as integration. And integration, this can get really kind of complex. We'll see some examples of this as we go through this class today. It can get really complex, and we will try not to get too complex with this, at least in our class. Okay, integration is when you can have 2 or more signals with opposite effects. Okay, so in this example here, we have signal 1 going to receptor 1 and signal 2 going to receptor 2, and the signals cause the opposite effect. So in this case, the concentration of X goes up, or the membrane potential is going up. For receptor 2, the concentration of X goes down, or the membrane potential goes down. The net effect of these two is the net effect is the interplay between those two. Okay, and this is also referred to as crosstalk. So if you have 2 different receptors and 2 different pathways that sort of combine or converge on the same physiological response, and maybe they have opposite effects, or they might even have a similar effect, right? The net response of that cell is going to be the sum from both of those receptors, or however many receptors are acting on whatever that physiological response is. Okay, and again, that is referred to also as crosstalk. Okay, so here are 6 different types of signaling that can occur through a membrane. Okay, number 1, and this is the first example we'll see, are G protein coupled receptors. In these, you have a ligand that binds to your receptor. And I should mention too, before I get too far ahead of myself, 1 through 5 of these are all involving membrane-bound receptors. Okay. Number 6, nuclear receptors, those are the ones that aren't membrane-bound. Okay. So back to G proteins, or G protein coupled receptors. Okay, you have that ligand binding the receptor that activates a G protein. That's why they're called G coupled or G protein coupled receptors. Okay, that then will either activate another enzyme or inactivate another enzyme, depending on the pathway. All right, so those are the ones we'll be covering today. Receptor tyrosine kinases, and we'll talk about the structure in the next lecture a little bit, but they are integral membrane proteins that have very large regions inside and outside the cell. When they bind their ligand, they activate the intercellular part that then phosphorylates another protein, and then that protein phosphorylates another protein, and you get what's known as a kinase cascade. Kinase is a word for a protein that phosphorylates something. 3, the receptor guanylyl cyclase, and we won't really see, I don't think we see examples of this specifically in this class, but it binds a ligand and it turns GTP into cyclic GMP. We'll see something very similar here in a minute with ATP to cyclic AMP. 4, gated ion channels. These are ion channels that are, in general, they're closed when they don't have a ligand bound. The ligand will bind, and then that opens the channel and allows ions to go through. Adhesion receptors or integrins. Okay, these are a little bit bit different. Thus, they're binding their receptor, or excuse me, their ligand can be very large, extracellular matrix. Okay, and that changes a conformation, and that change in conformation is transduced through the membrane into the cytoskeleton. Okay, and then finally, nuclear receptors. You have a ligand that it's acting not on anything in the membrane, but on a receptor that's in the nucleus. Okay, and then that either, in general, it can either express a gene, or it can block the expression of a gene. Okay, and I should mention too that the tyrosine kinases, their kinase cascade eventually acts on an activator or a repressor of a gene within the nucleus. Okay, so moving on to section 2, and these are the G protein coupled receptors. They're alpha helical, and they have 7 transmembrane helices. Okay, and that's we've already talked about one of these, even though we didn't mention that it was a G protein coupled receptor, and that was rhodopsin, and we'll see that that again today, but that was the protein that had 7 transmembrane helices. Okay, that's a very widely studied G protein coupled receptor. And that's shown here in red. Okay, so those are the 7 transmembrane helices. Okay, and this part here, this is the G protein coupled receptor. That's in red. That is the part that's in the membrane, and for the most part, it does span the entire membrane. Okay. The other part of G protein coupled receptor pathways, you have the receptor part, but then you have the G protein as well. And that's shown here in the 3 different colors. Okay. Those G proteins are membrane associated proteins. They're not integral membrane proteins. You would really consider them peripheral membrane proteins. They have a very small anchor that keeps them associated with the membrane. And they have, most of them, the examples that we'll see are heterotrimeric, meaning they have 3 separate polypeptide chains that are all different in sequence, and those subunits are denoted alpha, beta, and gamma. Okay, some G proteins can be single chains. Those typically are much smaller. Okay, so here we see this. The G protein is actually larger than the receptor part. Okay. Okay. The G proteins are also the reason they're called G proteins is that they bind GTP, and that's very important for their activity, which we'll mention here shortly. The G proteins mediate this signal transduction from the receptor to other target proteins. So they're sort of like a middleman, if you will. This is a cartoon depiction of a G protein. And I'm going to say a few words about the actual mechanism, I guess, if you will, of the G protein. They have the cartoony blob here is the protein. And these are sort of the localized domains of this protein. There's a switch 1 and a switch 2 on either side of what's known as the P loop. Okay, and the P loop, I'm guessing that stands for phosphate loop. Not exactly sure, but that is the region where the triphosphate moiety of the GTP sits. Okay. And then in the switches, on switch 1 you have threonine at position 35 that is hydrogen bound to that an oxygen in that gamma phosphate group, and in switch 2 it's glycine at position 60. Okay. The G protein is active when it has GTP bound, and it's inactive when it has GDP bound. Okay, and the reason why these things don't just bind GTP and then stay active forever is that they actually have an enzymatic activity in them, and that is for the cleavage of that terminal phosphate group. Okay, so a G protein actually will catalyze the cleavage of that phosphate group in the GTP, which then inactivates it. Okay, which is, you might think that's kind of odd too. These things are waiting for their receptor to bind something, then to activate them, just to cut that off and inactivate themselves. But that really provides a time limit on how long they're active. Okay, you don't really want something to be active forever. Like, just keep producing that same signal, even or that same response, even though the signal has gone away. Okay, so that's one way in which these G proteins turn themselves off. This is a crystal structure of one of the more widely known G proteins, and that's ROS. We'll see more examples of that later in other lectures. Okay, and this is, you don't need to know any of this, but here's what it looks like in sort of real life as opposed to a cartoon. We have switch 1 here in red, P loop, and then this is switch 2. Okay, so you have these some binding between switch 1 and switch 2 in that terminal phosphate group. Right, so here are a few crystal structures of different G protein coupled receptors. They are just their positions in the membrane here are shown. Okay, so they span the entire membrane. There's a extracellular part and an intracellular part. Okay, the extracellular part is where that ligand is going to bind, the ligand binding site, and then the inside part is where it's going to be interacting with the G protein. So here is the beta 2 adrenergic receptor, and this was the one we're gonna talk in more detail about. Okay, there's also opioid receptors are like this. Histamine receptors are also G protein coupled receptors. Okay, the G protein here is shown G sub S alpha, G sub S sub beta, and G sub S gamma. If you see the G sub S, that S stands for oh geez, I just had it. Stimulating? That doesn't sound right, but it's, I think that's it. It means that they go on to activate something else. In the other type of G proteins that you see would be inhibitory, and we'll see an example of one of those. Okay. So epinephrine, or also known as adrenaline, is a hormone made in adrenal glands. Okay, and those are organs on top of the kidneys. Okay, what epinephrine does is it mediates a stress response, and you've probably heard of the flight or fight response. And what that does, what this molecule does, it's a little bit of it's tissue specific, but what it's doing is it's going to be mobilizing energy sources so that you can use them to either fight off something or run away really quickly from some danger. In muscle or liver cells, this hormone epinephrine, it induces the breakdown of glycogen into glucose, right? So it releases a bunch of glucose. That glucose then can be used, in your muscle cells to run away really fast or to fight something off. In fat tissue, adipose cells, it induces lipid hydrolysis, which again gets free fatty acids available to be metabolized. In heart cells, it increases heart rate to speed up blood flow, to get those metabolites to different tissues, to get oxygen to your muscles quicker when you're running away from something. And what epinephrine looks like, the structure is shown here. It's a relatively small molecule, sort of a benzene ring with two OH groups, two hydroxyls, and then we have a four-membered chain on the other side of this benzene ring. Okay, and that, there's one alcohol group, an amine group in there, in that part of that chain. So nothing like, I guess nothing that really stands out drastically, but it does have a very specific shape, and it fits in that receptor, very well. And the KD value shown here, it's 5 micromolar. There are synthetic molecules that you can make as drugs. One of them is shown here. You don't need to know that structure, or the name of it really. What's important to note is that this is an agonist, and what agonist means is it's a molecule that binds in a receptor, and it gives the same response as the natural ligand. Okay? So this turns on that receptor just like epinephrine would turn that receptor on. The difference is, if you look at the KD, this synthetic agonist has a much smaller KD, so it actually binds in there more tightly. Okay? So you can design drugs that work better than the natural ligands, at least that bind better than the natural ligands. Okay. The other drug shown down here is what's known as an antagonist. An antagonist, you can also think of it as an inhibitor. It will bind in a receptor, but it blocks the natural response of whatever that receptor does. Okay, so this is an example of something that would bind and inhibit or block that signaling pathway. Okay, and that KD value is much lower than the KD value of the natural ligand. So there's another example of how you can purposely design something that fits even better than the natural ligand. Or I shouldn't say fits better necessarily, but it stays in that binding pocket longer. It's held tighter. Okay, let's look at this beta adrenergic receptor pathway in some more detail. Okay, and the beta adrenergic receptor, okay, that's another name for the epinephrine receptor. Okay. So that can be a little bit confusing. Adrenaline, epinephrine, and beta adrenergic receptor, we're all talking about the same thing. Okay. And I should mention here too that now that we're covering these signaling pathways, it can get a little bit complex, because this is really our first look at pathways, true pathways. And the thing about pathways that's can be a little bit tricky for some people, me included, is that they really do require some memorization. They're pretty much all memorization when you're going through a pathway. It's sort of where biology comes into play now. So I will try to highlight some of the important features of these. We'll go through it. Hopefully that helps you to remember them a little bit. But you probably do need to read through the book and look at these figures in some detail to remember them, and even writing them out helps a lot, like on note cards or something. I probably wouldn't, now if this was kind of a normal class and not online lectures, I could ask you to draw a pathway from scratch, but in general, I try to stay away from those types of questions, but they would be sort of fair game. On an online exam, I'm probably not going to ask you to recreate an entire signaling pathway, but explaining different steps or picking out examples of the 5 features of all signaling transducing pathways in these would be kind of fair game. Okay, so there's my spiel. So let's look at this one in a little bit more detail. So the first thing that happens, as you would expect, is that your ligand is going to bind to the receptor. So in this case, the beta-adrenergic receptor binds epinephrine. So epinephrine, here shown in this little cartoony kind of brown color, goes into the receptor, the beta-adrenergic receptor, and binds. Okay, so this is our G protein coupled receptor. It is 7 transmembrane helices, the binding pockets on the outside of the cell. Okay, that epinephrine binds. Okay, what that does is it causes the GDP that's bound in our alpha subunit, GS alpha, to be released. So that binds, G alpha will release GDP and it will bind GTP. Okay, when it does that, it separates from the beta and gamma subunits of the G protein, and then it becomes more mobile and it can move around. Okay, in the case of the beta adrenergic receptor, that G protein is going to move until it finds its target enzyme, known as adenylyl cyclase. Right. This active G alpha subunit, when it has GDP, will then activate adenylyl cyclase, bind to it and activate it. Adenylyl cyclase then will take ATP and convert it to cyclic AMP. And we'll see that molecule in detail here in a second. Okay, cyclic AMP is then a sort of a secondary messenger, if you will. It will then go to activate a protein known as pKa. You can think of that as standing for protein kinase A. And remember what a kinase is. A kinase is an enzyme that phosphorylates other proteins. So pKa then phosphorylates other proteins, and it actually has lots of different targets that it can phosphorylate. Okay, and so those other proteins that get phosphorylated then go on to give the cellular response to epinephrine. Okay. The last step here is that cyclic AMP is then degraded, and then that reverses the activation of pKa. And we'll look at in some of these steps in a little bit more detail here. Okay, this is crystal structures of this receptor system, and modeled in a molecular dynamic simulation with membrane molecules around them, just to show you maybe a little bit more how this looks in all actuality, not just a cartoon picture. Okay, but again, you don't really need to know any of these, right? This obviously wouldn't show up on an exam. Just a better idea of more a realistic picture of this. So cyclic AMP is a pretty interesting molecule. It's made from ATP, and that adenylyl cyclase enzyme cuts off the 2 phosphate groups and leaves an AMP, but it also catalyzes the formation of a bond between this first phosphate, the alpha phosphate, and the OH group on this ribose sugar in the ATP to make a cyclic structure. Okay, and so the cyclic structure, that phosphate's going to be bound to the oxygen on the 3' oxygen and the 5' oxygen. Okay, so that's cyclic AMP. We haven't talked about sugar structure, carbohydrate structures yet, so I wouldn't expect you um, to draw this molecule right now. We'll be talking about carbohydrate structure in chapter 7. So for now, I would kind of expect you, if I showed you a picture, you should be able to recognize this because it has this very weird um, cyclic structure with a phosphate. Okay. This stays active until another enzyme known as a nucleotide phosphodiesterase cuts that bond here at the 3', and then that gives 5' AMP. Cyclic AMP is a secondary messenger, as we mentioned. It allosterically activates a protein known as CMP dependent protein kinase A, which is abbreviated as PKA. So you can think of that as protein kinase A. Okay. Protein kinase A then leads to the activation of many other enzymes. Most of the goal from that activation goes to produce glucose from glycogen, but there are other, depending on the tissue, there are other responses that PKA is involved in. Okay, so in this pathway we see some examples of some of the features we talked about and all the general features, right? So amplification, we see this. Okay, a few of these G protein coupled receptors leads to the activation of a few adenylyl cyclase enzymes. Every active adenylyl cyclase enzyme makes several cyclic AMP molecules, and then those cyclic AMP activate several PKA enzymes, and then this is where it really gets amplified. The PKA enzymes then have a kinase cascade, and thousands of glycogen degrading enzymes, at least in liver tissue, are activated. And in the end, tens of thousands or hundreds of thousands of glucose molecules can be released into the bloodstream. And it's probably easier to see this with a picture. Okay, so we have one molecule of epinephrine binding to that receptor, right? That's going to activate the one receptor, right? That's one receptor that's being activated by one ligand. And then that can activate, just on this is sort of an estimation, but you could say maybe 10 of these G alpha subunits. 10 of those active G alpha subunits can then go on to activate adenylyl cyclase, or 10 adenylyl cyclase molecules. Those could take and produce maybe 200 molecules of cyclic AMP. 200 molecules of cyclic AMP would activate 100 molecules of PKA. And we'll talk about why that is, that the number actually goes down there. PKA, 100 molecules of active PKA, then can go on to produce 1,000 molecules of another enzyme. And these enzymes here, you really don't need to know their names, because we don't really, this is, I think, really the only place we see them. So you have a tenfold increase here with this step, and then this is another kinase, so it's going to phosphorylate another enzyme, and you get another tenfold increase. So now we've gone from 100 molecules of PKA in 2 steps. Now we're at 10,000 active molecules of this glycogen phosphorylase A. That then can break down glycogen into glucose, glucose 1-phosphate specifically, and you have 10,000 molecules of that can liberate 100,000 molecules of glucose. Okay, so that would be the example of signal amplification in the beta-adrenergic pathway. Okay, another feature that we see is feedback. Okay, epinephrine's meant to be a short-acting signal, so the cell needs to stop the and I don't like this word here, synthesis. It should, I think it should be glucose liberation or glucose release, because it's not technically synthesizing it. Glycogen is just a chain of glucose molecules. It's just cutting up glycogen into glucose, but just be aware of that. It's not actually synthesizing it. Okay, so the organism has to stop that production of glucose, that liberation of glucose, if there's no more need to either fight or flee. Okay, so downregulation of cyclic AMP occurs by the hydrolysis of GTP in the alpha subunit of the G protein. It is one way that feeds back. And we'll talk about another feedback mechanism as well. Okay, so this is our pathway. We've sort of gone through those first three steps, right? You have your epinephrine binding that allows the alpha subunit of the G protein to bind GTP and become active. You turn on adenylyl cyclase. Okay, so that's all well and good, but you don't want that to continue forever. So one way that is stopped is that there's that intrinsic time limit in that alpha subunit. After so long, it just goes snip, and it cuts that gamma phosphate off of the GTP, and it activates it. So that turns itself off. Okay, once that happens, it can then go back and associate with the beta and gamma subunits. Okay. There's also examples of integration here. Cyclic AMP is a very common secondary messenger. A large number of G protein coupled receptors mediate their effects via cyclic AMP. They can both be activating and inhibiting cyclic AMP synthesis. So you have some crosstalk there, right? Some of these receptors activate cyclic AMP synthesis, cause that concentration to go up. Others inhibit it and cause the concentration to go down. The human genome encodes about 1,000 G protein coupled receptors. The ligands are things like hormones or growth factors, neurotransmitters, so many different types of ligands for these receptors. There's also hundreds of different G protein coupled receptors that can be responsible for similar processes. And the examples there are things like taste and smell. And also, many of the ligands for these receptors have yet to be identified. You can go and do a sequence search of the genome, and very well these G protein receptors are 7 transmembrane helices. So you can match up the sequence of a known G protein coupled receptor and sort of map that and search the genome and find other very similar genes that have to be encoding other G protein coupled receptors. So by doing that, you can see that there are about 1,000 of them, and only I don't know the number exactly, but only a fraction of those that we know their receptor. There's some that we don't even know the receptor yet, the receptor molecule, ligand, I should say. Okay, so here is table 12.2, and this shows some enzymes or other proteins that are regulated by cyclic AMP dependent phosphorylation by that PKA enzyme. Don't need to know these by heart or memorize them. It's just really here to emphasize the point that pKa is sort of promiscuous. It will phosphorylate many different proteins. Most typically, it does it at serine, the amino acid serine. But here's one example, at least, of a threonine that it phosphorylates. Okay, the pathways they're involved in are shown here. Okay, so some of these enzymes that pKa phosphorylates will be more or less expressed in depending on what tissue you're in. And so then the end response will be slightly different in different tissues. Okay, and there's also, if quite a few of these are expressed in a single cell, there's going to be some crosstalk here, right? If pKa is phosphorylating all of these, right? The pathway that or response that gets turned on, there's going to be some sort of interplay here because these are all involved in different responses. Okay. Some signals that use cyclic AMP as a secondary messenger, okay, they're shown here. Right, we've seen the epinephrine would be the signal, the example that we showed. Others, maybe pick out some that might be important. Glucagon, we'll talk a little bit more about that next time. It's interplay with insulin. Histamine, having allergic responses involve histamine. What are some other good ones? Serotonin, the neurotransmitter, and even like thyroid stimulating hormone, TSH. So there's many different pathways, signaling pathways that use cyclic AMP. And this list probably would be the better example of crosstalk, because these pathways are going to have, in some cases, some of these pathways are going to have opposite of physiological effects on a cell. Okay. Cyclic AMP is able to mediate multiple signals because of its location of the localization of protein kinase A. So protein kinase A, it's not really just floating around randomly. It is in close proximity with the adenylyl cyclase and a beta adrenergic receptor via these protein complexes, and this is really more complicated than we need to get in this class, but you can think of it as pKa is very, very close proximity to adenylyl cyclase, and that's why right, we typically in the other figures we showed adenylyl cyclase making cyclic AMP, and then that goes off and finds pKa somewhere, but in reality it's very close to adenylyl cyclase, so as soon as cyclic AMP is made, it can go and activate pKa. Okay, so how is that activated? Well, it turns out that pKa is a tetramer, and it's actually the tetramer is a dimer of two heterodimers. It's a dimer of a dimer, which makes up 4. Okay, so if you just focus on one part of this tetramer, so just take this top half. This is pKa. protein kinase A. Okay, it has one dimer. The top dimer has an R subunit and a C subunit. Okay, the R stands for a receptor subunit, and the C stands for a catalytic subunit. Okay. So there's one of those on top, one of those on bottom. Right. It turns out that you need two cyclic AMPs to bind to one of the receptor regions to release the catalytic site. And when that catalytic site subunit is released, the catalytic site is exposed and it becomes active. Okay, so in this case, for the tetramer, because there's two R sites, you're going to need 4 cyclic AMPs. Okay, so 4 cyclic AMPs would then go on to release 2 active pKa catalytic sites. Okay, that is the reason when we are talking about the signal amplification, that step where between cyclic AMP and pKa, we actually went down by a factor of 2. That's why, because we need 2 cyclic AMPs to activate 1 pKa. Right. Once those pKas are active, what they do is they take ATP and they stick it on different proteins. They phosphorylate, they take a phosphate from ATP and stick it on, remember most commonly it's serine, but sometimes it can be threonine. They phosphorylate proteins at those amino acids. Okay. It turns out that one of the proteins pKa phosphorylates is phosphodiesterase, PDE. Phosphodiesterase, if you also remember, that is the enzyme that cuts the cyclic AMP and converts it back into AMP, just normal AMP, thereby inactivating the cyclic AMP. So if you have these pKa molecules, they're active, you have enough of them around, eventually they're gonna go and they find PDE and they phosphorylate it, which activates it. Then PDE can cut some cyclic AMP and in a sense get rid of the cyclic AMP, lower that cyclic AMP concentration, and thereby inactivate pKa. So this is a great example of feedback. The feedback inhibition that will actually turn off this response in the beta-adrenergic receptor pathway, and really any pathway that involves cyclic AMP. Some bacteria toxins are enzymes that target G proteins. And examples are from cholera and whooping cough toxins. Respond in this way. They really what happens is they modify the G protein, the alpha subunit of the G protein, in such a way that it always activates the adenylyl cyclase, and then adenylyl cyclase is always on, and you're going to be producing too much cyclic AMP from ATP. Okay, and that uses up ATP, which isn't good. And then it also, you have a lot of cyclic AMP floating around, which then causes a lot of different physiological responses that don't necessarily need to be happening at that time. Okay. These G protein coupled receptors can also use other secondary messenger molecules. And an example here is what's known as inositol 1,4,5 triphosphate. I think we saw this was one of the lipids we talked about, head groups. You can also just think of this as IP3 or simply inositol, I think would be fine. It's typically in the form of phosphatidylinositol 3-phosphate, and it would be in the membrane. The head group is that inositol 3-phosphate. Okay, when the G protein, when a hormone binds in this receptor, turns that G protein on. That G protein then activates what's known as phospholipase C. That cuts that head group off of that phosphatidylinositol, and then that inositol 3-phosphate will then go and bind to a ligand-gated ion channel, which then lets calcium into the cell. Then calcium binds to protein kinase C, and it activates it for further response. This pathway, you don't need to know the specifics of this pathway, just that there's other molecules other than cyclic AMP that can be used as secondary messengers. Okay. All right, so we're going to move on to our second example that we'll see in some detail of a G protein coupled receptor. Okay, and that is in sight. And in sight we have rods and cone cells that then transduce I don't like this. I really feel like the light should be coming in this way and hitting the rods and cones. And then that elicits a response from into your neurons and into the nerve cells, which then goes to the brain and depicts that sight, the light hitting the rods and cones as an image. Okay. Right, so what happens in here? This is really a sort of an FYI, but what we have is that we have a molecule called cyclic GMP, right? Very similar to cyclic AMP, but we have a different base on that nucleotide, right? We have a G instead of an A. That is a ligand for a ligand-gated ion channel, and when that is bound, the ion channel is open. Okay? When the ion channel is open, what we have is influx of calcium and sodium into the rod, and then we have also this sodium pump pumping sodium molecules out, right? So you're actively pumping sodium molecules out so that you can use this SIM port. This ligand-gated ion channel acts as a SIM port. The sodium will move down its concentration gradient and bring a calcium against its concentration gradient. Okay, so this would be an example of secondary active transport. Okay. So that is producing a particular membrane potential shown here. Okay, when light hits this cell, this rod cell, what happens is the cyclic GMP is not bound to that ion channel, then it's closed. You don't get influx of sodium or calcium in, right? You're getting a sodium out, right? And then the membrane potential becomes lower, and then that lower membrane potential is causing a electrical signal, which is then transported to the next neuron, and then finally through nerve cells all the way into the brain. Okay. The G protein coupled receptor involved in this pathway is known as rhodopsin. Okay, and rhodopsin is shown here in red with the 7 transmembrane helices. It's a little bit different because its ligand is is sort of already bound there in that receptor. Okay, it's not, and this is a receptor for light, right? So it has its ligand kind of, I don't want to say permanently bound because I'm not 100% sure if it can come out and go back in, but we can think of it as being permanently bound in there. Okay, and then when the light hits the ligand, that ligand changes shape, and then that transduces the signal to the G protein. Okay. The G protein in this particular example is known as transducin. Okay, so this is the G protein involved in sight, and again, it's a stimulatory G protein. So we refer to it as G sub S. And here's a little bit better picture, I think, of these G proteins have a little anchor here, in this case, isoprene groups that keep them associated with the membrane. All right, this is our pathway for our, we can just call this a site pathway or the rhodopsin pathway. It's shown here. There are two different parts to it. Okay, one is the excitation part, that is, the light hitting rhodopsin, and then the G protein being activated, and what happens through that pathway, through that signal transduction that ultimately leads to the cyclic GMP concentration to be lowered. Okay, that's all the excitatory phase. There's also the other part of that, right? Once that happens, and you send a signal, you have to be able to recover from that, and get back to your starting point, so it can then sense another photon of light. Okay. In this class, you only need to really focus on the excitation phase. Okay, we're not going to focus on the recovery phase. If you're curious about this, like you think this is going to be on the MCAT or something, or you're just interested in it, by all means, feel free to study that part. But just to simplify things a bit, we're going to focus only on the excitation phase. So on an exam, I'm not going to ask any questions about the recovery phase. Okay. Good. So looking at this in a little bit more detail, the first thing that happens is light is going to hit this rhodopsin molecule, and when that happens, visible light, it's going to change that ligand that's in there, and it's, if you remember, we've talked about this before, it's 11-cis retinal is that ligand that's in there. When light hits it, it becomes all trans or trans retinal. Okay, and that change in conformation of the ligand then changes the structure slightly of that receptor, and that's conduced into a structural change into transducin in the alpha subunit. GTP can then bind, which activates transducin alpha, okay, which is again the G protein. That then goes and activates the enzyme PDE, right, phosphodiesterase, and we've seen that. And how it activates it is kind of a little bit unique. Phosphodiesterase has an inhibitor bound to it, an inhibitory subunit, and what transducin does is it takes that inhibitory subunit away, so that inhibitory subunit on PDE then binds to transducin alpha, and PDE becomes active. Okay, and this PDE, and you can think of this PDE, it's going to do the same thing as it did to cyclic AMP. Remember, we talked about PDE once it's activated by protein kinase A, it it um, it it starts to cut cyclic AMP and makes AMP, right? So that this is a different PDE, and this PDE is specific for cyclic GMP, but it does the same thing. It takes cyclic GMP and it converts it into 5' GMP. So it cuts and breaks the ring in the cyclic GMP. Okay, as it's active, it'll do that, and eventually it lowers that concentration of cyclic GMP. When that concentration's low enough, right, that the cyclic GMP is not going to be in that ligand gated receptor, and those are going to close. Okay? And those closing means that you're not going to get the calcium in there, and the membrane potential is going to be what's known as hyperpolarized. Okay? And then the polarization, the hyperpolarization of that membrane is what then sends a signal to the next the neuron, and then that signal is then propagated all the way to the brain. And again, that process of sending the signal to the next cell, those can be very complex processes. So as far as we're concerned with sight, this is sort of what you need to know about it as far as the signaling pathway. Okay. Other sensory perception is mediated by these G protein coupled receptors. Vasopressin, this is a hormone or signaling molecule that really has the opposite effect of epinephrine. Okay, so these two would be examples of things that are acting on the same sort of physiological responses, but in opposite ways. Okay, their vasopressin actually has a G protein with the sub-I. So that's an example here of an inhibitory G protein. So when vasopressin binds to its receptor, the inhibitory G alpha subunit is then liberated, and it goes and turns off adenylyl cyclase. Okay, and what that does is it lowers the concentration of cyclic AMP, and it inactivates, in a sense, PKA. Okay, right? Epinephrine does the opposite. So these two, vasopressin and epinephrine, if you have both of those signaling molecules around at the same time, you're going to get that would be an example of crosstalk or integration, right? The net effect will be the interplay between those two. Right, we've talked about this light. Obviously, that's sensory. Odorants are also using G protein coupled receptors, and so they can do that either in that very quick example we saw of using a lipase that releases the inositol, which then binds to a ligand-gated ion channel. They can use that, or they can also use cyclic AMP to do the same thing with an ion channel. And then taste also can use G protein coupled receptors. Okay, you have another interplay with the adenylyl cyclase and cyclic AMP controlling a ligand channel that's controlled by being phosphorylated. Okay, So you see that there's, many of these pathways are using, at least three of these that are shown use cyclic AMP. So really, it, the the overall physiological response is going to be dependent on the tissue type, because some tissues might have, in your tongue, right, you're going to have more of these receptors for taste molecules, or in your nose you'd have receptors for odorants, and you might not have as many receptors, right? You definitely were not going to have a rhodopsin receptor in your nose, right? So the interplay becomes more tissue specific based on what proteins are expressed in that specific tissue, and what proteins need to be expressed to give you the specific physiological responses you need. Okay, so that is all we have on G protein coupled receptors. Next time we're going to talk about gated ion channels, which comes from section 6, receptor tyrosine kinases, which comes from section 3, and then a little bit about cell cycle regulation, and that's section 11. And we talk about that because that is sort of a more of a specific example of receptor tyrosine kinases.